In this video, I want to begin talking about what it is that DNA is doing in the cell. So we've learned something about DNA. We've learned something about how DNA is replicated in cells. And now let's talk about, well, what it's actually doing. So what do we already know about DNA and RNA? We know that they are both nucleic acids and that they're composed of 4 nucleotide bases. If you remember from hopefully learning this at some other time, in RNA, it's a single strand rather than a double strand. And there's a different nucleotide in RNA. Instead of thymine, we have uracil. So wherever adenine is binding, it's not binding to thymine, it's binding to another nucleotide called uracil. And like I said, it's a single strand, not a double strand. And then finally, it's got a different sugar. RNA has ribose sugar instead of deoxyribose sugar. So those are the differences. Now, DNA, we say it's the molecule of the cell, it's the molecule of life, it's containing all the information for living things, but really what it is, literally, is the code for making a protein. So what we call the central dogma of biology is DNA contains the code to make RNA, specifically mRNA, messenger RNA, and that process we call transcription. And mRNA leaves the nucleus and directs the process of protein synthesis to make proteins of all kinds, and you should remember that there are many different kinds of proteins. And then that process is called translation. We often use the term one gene, in other words DNA, to one polypeptide, that is protein, to describe the relationship represented in the central dogma of biology. But it's not as simple as that. So maybe you learn that in biology or honors biology, but of course in AP biology, there's more to it. It's not as simple as that. In eukaryotic cells, the gene transcribed from the DNA is altered before it's translated. It's altered. So the mRNA is transcribed from the DNA, but then there's going to be some things that happen to it before it's able to be used to make a protein. This processing or alteration happens in the nucleus. The initial transcription process from DNA to mRNA happens very similarly to the way that DNA replication happens, except it's using an enzyme instead of DNA polymerase. It uses an enzyme called RNA polymerase, very slightly different enzyme. This initial transcription product, the mRNA, the original mRNA that comes from that process, is called pre-mRNA. So the process might look something like this. We have the DNA within the nucleus being transcribed into something called pre-mRNA, and then there's processing. We call it mRNA processing. Notice it's got a green ball and a blue tail on it, and we'll talk about what that is. Then it's going to leave the nucleus, go to a ribosome, and direct the process of protein synthesis, which is, of course, what the whole intent of it is with DNA. So first we'll discuss what happens, and then we'll talk about why afterward. The first thing that happens, the first alteration of the pre-mRNA, is an addition of methylated GTP as in guanosine triphosphate, similar to ATP, adenine triphosphate, or adenosine triphosphate. So it's a similar molecule to that, but with a methyl group on there. Remember what methyl is. It's a CH3. That's methyl. So there's a methylated GTP molecule added on, and it's added to the 5' end of the transcript. In other words, the mRNA that's been transcribed, it's on the 5' end, because remember, RNA is similar to DNA. It's got a 5' end and a 3' end as well. So we call this the 5' cap, or the methyl cap, because it's on the 5' end is considered the head of the DNA molecule. So that's why it's called that. So that looks something like this. On the head end, so to speak, of the mRNA is where you'll find this 5' methylated cap. Looks something like this if you were to actually look at it. So here and here are some nucleotides. Then we have attached to it these 3 phosphates, just like in ATP, the 3 phosphate groups. And then you've got this 7-methylguanosine with the methyl group here. So that is your 5' cap that's attached on the end, the 5' end of the transcript. On the other end, on the tail end, so that's going to be the 3' end, is the addition of multiple adenines. Okay, it looks like here. So at the 3' end, there's a series of between 100 and 200 adenine nucleotides added. It's called the poly A tail, the polyadenine tail. And so on the 5' end, you're going to have the methyl cap, and then on the tail, the 3' end, you have a whole bunch of adenines added to the end, poly A tail. Then the RNA is spliced. Okay, so bits are cut out and bits are spliced together. This is the final step in the processing of the RNA. It's called RNA splicing. During splicing, portions of the original transcript are clipped out. The portions that are clipped out are called introns, which means intragenic, so they stay in. They stay out. And the portions retained and spliced together are called exons because these are the ones that are going to be expressed. It's a little bit backwards when you think about it because usually in means stay in and X means stay out, but it's opposite in this case because of the terms that they were originated from. So introns are clipped out and exons are spliced together to create the completed mRNA molecule. So the completed mRNA has the methyl cap, the poly A tail, and then it is made up of linked bonds. So something like this. Here's our pre-mRNA. In this case, we have a bunch of introns in there, the orange introns, and you've got, in this case, a 5' cap already and a poly A tail already, and then during the process of RNA processing, those little orange intron areas are clipped out. The exon green areas are pieced together, and now it's a completed mRNA, which can go direct the process of translation or synthesis. Now, how does that happen? Soon after the RNA is transcribed, it's bound or held together by small nuclear ribonucleic protein complexes, which is a big mouthful, but we abbreviate them SNRPs, SNRNPs. Small nuclear ribonucleic protein complexes, we call them SNRPs. These are proteins, and these SNRPs are made of protein, like I said, and then nucleotides, and they're responsible, they do the clipping. Okay, they're like the scissors, they can clip it up. The SNRPs bind to specific base sequences on the pre-mRNA. They recognize certain sequences on the pre-mRNA, the nucleotide sequence, and those are called consensus sequences. Those are frequently found in the genome, but they recognize certain ones. There are different kinds of SNRPs that recognize different ones, and then once they come in and bind, the SNRPs form a large complex that's called a spliceosome. A spliceosome. So there might be 3 SNRPs or 4 SNRPs or sometimes many SNRPs or sometimes just a couple of SNRPs, but they come in, they recognize a certain sequence in the mRNA, they bind to it, they create something called a spliceosome. On one transcript, there may be many spliceosomes that splice out many introns. Okay, so there might be 100 introns clipped out. A given gene sequence may have as many as 50 introns. In the final step of the process, the 2 ends of the exon are spliced back together, and the intron loop is removed and degraded by enzymes. So, go back. Something like this. Okay, so you have the intron here in the middle that's orange, right here. And then you have these SNRPs coming in, these green SNRPs. They bind together, they recognize the sequences on either end of the intron. They form this spliceosome, which loops that intron, and then they splice it. In the process of clipping out the intron, they also glue together the 2 exons to form the completed mRNA or mature mRNA. So something like this, where you have the SNRPs coming together to form the spliceosome, and then eventually gluing together the 2 pieces of it. And again, here it is again, a different way of looking at it. There might be 2 of these SNRPs, there might be 3, they might be 5, they might be 8. I'm going to skip that. So why is the mRNA not equal in length to the DNA it was transcribed from? It's not equal in length because introns are clipped out. Now, let's talk about why. We'll discuss why these modifications occur. Why do eukaryotic cells go to all this trouble? It seems like a lot of work. Why not just use the DNA as is? Well, let's talk about the 5' cap. The 5' cap facilitates attachment of the final mRNA to the ribosome during translation and stabilizes the mRNA. So without that 5' cap, it cannot properly attach to the ribosome and cannot be properly transcribed. So that's an important role. The poly A tail has a very important role. It assists in transport of the mRNA out of the nucleus, but it also helps prevent degradation. So if degradation of the DNA happens, and we kind of talked about telomeres, it's sort of related to that. over time it might get degraded. It's going to be out out of the nucleus in the cytoplasm. There are enzymes around that could be potentially harmful to it. So if there's going to be degradation, it's going to happen there, and it'll start cutting off those adenines, but it's not going to hurt the transcribed section, if that makes sense. Those extra adenines are the ones that get damaged, which doesn't affect the making of the protein. How is an intron recognized so that it can be cut out during splicing? Well, the presence of introns in the genome is in part due to genes from our evolutionary heritage, and they were never removed. So it's junk DNA, they call it. It's just extra DNA that's in there. But because it requires so much effort to remove them, it is likely that they actually have a function. It's not just junk DNA. Because they're still in there, even though they're junk DNA, why wouldn't over evolutionary time they have been removed? Well, it's likely that they have a role. One function might be to provide extra variation, especially in small populations. They also provide a way, and this is so important, to code for more than one polypeptide with a single gene. It's genius. Of course, life is always genius, but this provides for alternate patterns of splicing, giving different products, in other words, different proteins from the same section of DNA. It magnifies the amount of variation you can get from a single section of DNA. And usually these are proteins that are related. So alternate splicing gives you sort of similar proteins, but just with a slight change. With one signal, a certain intron is snipped out, and then with another, it is left back. So if we look at that, if we start here with our DNA molecule, and this is the whole segment, then we have our pre-mRNA, which is a copy of each of these little sections. And then after mRNA processing, in this one, the green was clipped out. In this one, we left them all in. And in this one, the red was clipped out. So after translation, we get 3 different proteins from that same section of DNA. Okay, that's called alternate splicing or alternative splicing. What will be the products of the splicing of the following pre-mRNA molecule? That's it. So that's an example. So we have our 5' cap, we have our poly A tail, and then we're going to clip out this intron so that we end up with the 2 exons spliced together. And I believe that might be the end of my lesson. So that was it for mRNA processing.